Okay, please go ahead. You can start.
Okay. Um, Thank you. So I would like to again say hello to everyone. So today we are meeting for this webinar to learn more about biomedical data science and drug discovery. So this program will introduce participants to the application of data science to important challenges in clinical and pharmaceutical application, especially, and we will be using practical examples that will cover genomics, transcriptomics, and structural data. So this collaborative program is designed for students by Pine Biotech, USA, Pharma Academy India, Pharma Toppers India, and Probiotechnica India. So before we let you know more about what we will be covering in the session, just to learn about why we are doing this. So right now we live in a world that is recording data every day. So the healthcare data related to clinical trials, electronic medical records such as EMRs, and then genetic information, then care management databases, then you have billings, and then you have internet research and social media, all sort of data. So the data that is related to healthcare is growing massively, and at the same time it poses a challenge to the community of students, scientists, and also researchers. So this challenge is also an opportunity to learn and discover from this vast amount of information that is now available publicly. So that is one of the main purpose of doing this program. So after working for the last 8 months with a group of experts from academia and industry leaders, we were really excited to bring this program to the students, where you have an opportunity to learn about different omics data types, and then relate and then learn from the examples that how this can relate to the real data driven drug discovery pipeline, and learn about these diseases and indications. So this summer program that is being conceptualized by our partners and Pine Biotech. So to tell you more about Pine Biotech, so Pine Biotech is a US-based company that provides resources, solutions, and training for research and education in biomedical informatics. So we have been developing an innovative approach to incorporate bioinformatics training, educational materials, and biomedical specialization for universities, companies, and top research labs around the world. So our team is working with academic and industry partners around the world to adapt and utilize our training resources for the need of universities, professional development programs, and also for school education. And to tell you more about other partners of the program, Faba Academy. Faba Academy is a special wing under Faba organization that focuses on grooming the students of the university nationwide and make them future ready. And about Pharma Toppers. So Pharma Toppers is a pioneering institute committed to provide refined education to pharmacy aspirants for preparing for postgraduate pharmacy entrance exams, as well as giving them opportunities to learn from data science and application of data science in chemoinformatics and application of data science in biology. The other partner is Probiotechnica Research Private Limited. So the Probiotechnica Research Private Limited is on a mission to equip and empower every students and faculty with latest technologies and tools to create an edge from others. So the company strongly believes in serving society and quality work. So for this program, all of us have come together to bring you best of the resources, best of the opportunities to learn about biomedical data science and drug discovery. So the second phase of the program, where we are going to start learning about a very important data type, that is transcriptomics data type. So let me tell you a little bit about this transcriptomics data type. So in the last decade, transcriptome-based analysis has been one of the most utilized approach to investigate human diseases at molecular level. So, I mean, you can understand the importance of this technique in human health. So, through these many expression studies, many molecular biomarkers and therapeutic targets have been found for several human pathologies. And to talk more about omics logic education, research, and the application of gene expression in biomedical research, we have none other than our CEO, Ilya Brodsky. So I'd like to now next invite Ilya to talk more about the application of gene expression in biomedical research. Over to Ilya.
Okay, thank you, Mohit. And thank you everyone for joining today's session. As Mohit already mentioned, I will be speaking a little bit more about this data type, what transcriptomics is, and what we will be covering in this upcoming program. But I also want to give you a sense of what kinds of practical assignments and tutorials you will have access to, and show you a little bit about how this program is going to work. So as we are getting started, and before we jump into transcriptomics, I want to ask all of you to log in to our Omics Logic portal, because we will be using this login to show some demonstration later in the session. So you can go to learn.omicslogic.com. Mohit, maybe you could post that link in there. So this is what it looks like. This is a portal that combines everything that you need to start learning about bioinformatics, starting from conceptual understanding of the logic behind different data types and their analysis, to big data processing tools that you can leverage to actually analyze your own large data sets and integrate them together, learning coding and different statistical and machine learning techniques so that you can manipulate your data and extract meaningful insights. And then also introduces you to a number of curated projects. Some of them we'll be going through in this program. So once you are registered, and you can let me know in the chat box, thank you, Mohit, for posting that link. So once you have registered, the way you register here, it's a free registration, right? You don't need to pay for anything. You just go to log in. Here you will see I can already log in because I have an account, but you can create an account right here. And it's just a very simple sign up. You can also use one of your social media platforms right here to log in and create an account. But once you log in, you will be able to see some of the resources that we'll be utilizing in today's session, because we wanted to make it a practical experience for you to be able to try some of the things that we'll be doing in the full program. Okay, so please let me know once you have registered and signed in. I will be just giving a couple of minutes for everybody to complete this step. And once you are logged in, you will see that you can complete your profile if you wish to do so, and some of the resources, I will be speaking about them in just a second. So please let me know once you're in. You can let me know by putting number 1 or logged in, signed up, or something like that in chat. And that way I will know that some of you are already registered, and I'll be able to move on. Okay, so I see that approximately 10 of you have already signed up. That's great. Okay, please let me know once you're all signed up. And all right, thank you very much. Okay, so what is this portal? The Omics Logic Bioinformatics Training Portal is currently serving over 10,000 users in over 30 universities across 5 different local chapters. And so we have representatives in the US, in Israel, Sri Lanka, India, Nigeria, Mexico, Ghana, Mexico, and others. And so we're very excited about recently putting in a lot of effort to redesign a lot of the materials that we've been using, because we believe that this kind of bioinformatics training should be accessible to people around the world. And hopefully you will appreciate how this works in today's webinar. Okay, so I see that many of you have signed up. Great, thank you. So the Omics Logic portal provides some free resources that you can get started with. And so after today's session, you'll be able to navigate through the different resources that we have. They include some training materials, data analysis tools, curated project data sets, and interactive tutorials that we'll be leveraging in the sessions in this program. And the way that this program is structured is we will have sessions just like we're having today. We will join Zoom. We have a communication platform on Slack dedicated to this particular program. So there you will have access to all of the members of this community, as well as our mentors that you can interact with, ask questions. There are some self-guided study materials that we'll be leveraging in between sessions, because we won't have too much time to do all of the practicum together. But you will get assignments. And then during the sessions, we will meet again and review some of those practical assignments, which will be done either on the T-Bioinfo platform, which I will introduce you to. So everybody that has created an account on the learnomicslogic.com portal will have a demo account today that you can start using to kind of explore the platform. But also we will have a lot of practical assignments using R and Python, and I will show you how that works today as well. Now, what we hope and our whole objective for the learnomicslogic.com portal is to provide participants an opportunity to develop independent research projects. And the reason why this is so critical is because I believe that the education system around the world is focusing on providing a lot of theoretical value and not enough comprehensive project-based learning. And so this portal is focused on project-based learning, and we have been privileged to work with hundreds of students that have developed independent research projects using their own data sets from public domain data, from NCBI, TCGA, and many other different projects. All right, great. Thank you everyone for joining. Now, just before we move on, I also want to mention that I know that many of you are likely joining our session today because you saw something on social media. And we have a group on LinkedIn where we post a lot of opportunities for internships, jobs in the pharmaceutical industry, in research as well. And we have a lot of experts that are actually able to provide mentorship and guidance to some of you that are interested to develop your career. So before we move on, I do want to mention that this is a perfect opportunity to share with you this group of professional students and researchers that you can interact with on LinkedIn and build your career. I also want to mention that we have a group on Facebook as well, also called Omics Logic, Data Science, Bioinformatics, and Biomedical Research. This group also has a lot of participants from around the world, over 2,000 members. So you're welcome to join that program as well. And we regularly post there a lot of information about resources, projects, and other types of opportunities that are coming up. Okay, so in this program, I am going to be sharing a little bit of a background information. Okay, so the Slack channel, we will get to that in a second. We are actually going to go through the registration process at the end of today's webinar. But before we do that, I will kind of share a little bit why we decided to make this program available online. So for the past probably 3 to 4 years, we have been working with many universities around the world, offering in-person training to students, faculty, and professionals in bioinformatics, data science, and other elements of data analysis. And these interactive sessions that we saw work so well online, we wanted to kind of translate that experience to online, where people could benefit from a practical experience, gain some skills, but also develop research ideas and translate those research ideas to real research projects. And so as a result, we have experience working with hundreds of faculty, researchers, clinicians, and students. And so what we are excited about is the opportunity that big data provides for people with all kinds of backgrounds, including engineering, biotechnology, biochemistry, biology, and life sciences in general, to be able to leverage this wave of big data that we are living through today. And so, as you probably all know, and Mohit already mentioned this, big data in biomedical research kind of became a thing around 2015, 2016. So a lot of people started thinking about big data, the problems and the opportunities that they have brought to the scientific community primarily, but also business-wise. And so many companies, as a result of seeing this opportunity, started developing tools and resources to integrate data and allow for user-friendly processing of data. But I think what we have all kind of left aside is that at the center of this kind of data analysis, integration, and decision-making stand people. And so our company is very highly focused on this part of this data analysis challenge. And this is really to bring in more people to be able to use these big data processing tools and resources and make sense of this data to be able to apply them in a meaningful way. And when I say all of these people, I really mean that we need to cover people that are lab technicians that need to generate more and better data. We're talking about people that develop new algorithms and new methods of analysis that need to understand where the data came from and who it's going to be used by. And we need to empower people that make decisions, which are clinicians and management that need to make precise decisions about what to do with the data, knowing the whole story of data generation, processing, and analysis. And so that's why we provide this Omics Logic training as a user-friendly experience that you can enter into and understand the logic behind analysis, and then learn about the specific tools that you need to use in a research project. Okay, so let me talk a little bit about why we are talking about transcriptomic data in the context of drug discovery, drug repurposing, and personalized medicine. So as you know, bioinformatics plays a critical role in modern drug discovery, repurposing, biomarker screening, and design of personalized treatment regimens. And so many industries and research labs are actually seeking out experts that are knowledgeable in omics data processing, analysis, and integration. And there are many different challenges as well as opportunities in being able to integrate, analyze, and turn this data into a model that could be used to design further experiments. Now, a lot of this is based on omics data, and the term omics actually means a lot of different types of data. And so this program is going to be focused on transcriptomic data, but the program that we are a part of today is covering several different elements of data that I think are important to understand. So one example is phenomics, and this means phenotypic data that can include cell morphology, the way that cells shape, right? Different cells have very different shapes, and they are dysregulated in different ways. We can also talk about genomic data, right? So the sequences of nucleotides and amino acids, typically referred to as genomic data. Epigenomic data, which is a field that has been growing very fast since about 2007, 2008, where we see an increase in the appreciation of how translation of sequences to mRNA and proteins is an important component of non-hereditary regulation of genomic sequencing data. And finally, what we will be focusing on in this program is the transcriptomic data. And so this program on transcriptomic data analysis is an important part because it provides a real in-depth view of how we can use gene expression and isoform expression patterns for discovery of biomarkers, for understanding of drug targets, and for being able to perform analysis of drug function based on expression of mRNA in different forms. So in this program, which is called RNA sequencing for drug discovery and repurposing, we will look for how people use transcriptomic data to link targets to diseases, validate these targets, and explore their variation in the natural systems biology. And so I think that this really brings into the spotlight this new era of drug discovery, where transcriptomics can be used for discovery of new compounds and complex molecules like proteins that can control disease through the regulation of gene expression that essentially mimic the mechanism by which our bodies exert regulatory control over a range of normal and disease functions. And so to speak a little bit about why next generation sequencing and what this term means and how we can use specifically this kind of data, let me briefly talk about the history behind next generation sequencing and other forms of measurement for mRNA. And this will help us see why we can actually use not just any type of mRNA measurement tool, but specifically next generation sequencing. So as a method of measuring RNA translation, transcriptomic studies can help detect mRNA expression inside the cell as an accurate alternative to membrane protein expression detection, which allows for molecular precision in diagnostics and even development of treatment based on molecular pathways. So what you can see here is the expression of the HER2 surface protein, which could be a measurement, a detection of that protein essentially tells us something about the property of the cell and about the processes inside the cell or subcellular molecular processes that are so important for understanding of that tissue condition and potential response to drug treatment. So when we think about our ability to detect such protein surfaces, there are actually several different techniques to do that. Before we get to RNA sequencing, we can talk about one of these technologies, which is microarray technologies. So the microarray technologies allow us to hybridize sequences of mRNA, so actual transcripts in these flow cells, that then we can extract RNA from cells or tissues and then see which ones bind together to these small sequences to measure abundance of specific molecules that we are interested in. Now, as opposed to microarray, next generation sequencing actually has revolutionized the way that we can look at transcriptomes because they provide us with so many novel insights about what's actually going on. One, we don't have to perform a preparation technique that creates the assay. We just measure all the mRNA that is present inside the cell, and then we can analyze what kind of alternative splicing events are happening. So what types of isoforms are being generated, what types of genes are being expressed, and then how can we combine or look separately at these two different types of data to make informed decisions about the types of samples that we are looking at. So what we'll talk about in this program is our ability to leverage this kind of big data and see the significance of mRNA expression in the context of data integration, which is really the frontier that drug discovery, drug repurposing, and personalized treatment deals with. So what's important here is that you will find many interesting and important biomedical projects that can leverage RNA-seq data for diagnostics and cancer biology understanding, for disease classification and patient stratification, which are so important for clinical trials and understanding of real patient populations that need to be treated, as well as single cell and tissue composition. So we will be able to look at how single cell and different types of immune cells, different types of tissues actually represent a heterogeneous composition of cells with different properties that need to be deconvoluted for us to be able to apply meaningful drug discovery and screening, as well as biomarker and precision medicine targeting of novel treatments. So here are a couple of examples of how people have reported on ability to use RNA sequencing and big data for drug repurposing and personalized treatment. So here you can see how, based on our understanding of specific patterns of mRNA expression, we can integrate data with phenotypic data, right? So essentially we take omics data and we combine it with phenotypic data. And based on our understanding of the way that these treatments actually work, we can then design better treatments or similar treatments for drug repurposing. In this example, you can see how this type of big data approach has been used to speed up the translation of treatment to the clinic from the starting point, not in the lab, but skipping through the lab and actually using this big data to identify important targets and similar conditions that could be targeted with existing drugs. And so those are some of the primary things that we will focus on. Data-driven discovery using big data sets that are available in the public domain, repurposing of existing treatments to treat conditions that are very similar to other ones based on their mRNA expression, and patient classification, where we can look at different types of populations that could be stratified for more effective treatment. The way this program will work is it will cover these 6 important topics. We will talk about tools and resources for transcriptomic data analysis. So we'll talk about what this data represents, how is it generated, and how to process so that we can have structured data that we can work with. We'll then look at processing of next generation data using bioinformatics pipelines. So how do we build pipelines? What are these pipelines? What are the steps in these pipelines? How do we perform pre-processing, mapping, and quantification? Then we'll talk about analysis, normalization, and visualization of gene and isoform expression. Then we'll talk about introduction to big data mining, clustering, machine learning, and predictive analysis. We'll talk about analysis of single-cell RNA-seq data for tissue-specific expression, deconvolution from bulk RNA-seq, and specific phenotyping of cell types. And then we'll talk about how to apply all of these tools in a meaningful way to a number of curated data sets. So we'll show you some examples, which I'll talk about today, that you can actually look through. Now, importantly, I'm sure that many of you have experienced this overwhelming feeling when you're coming to a new topic and somebody tells you that, oh, you can learn this in about a week. And you're thinking, I don't know so many elements of what you're talking about that I don't understand how all of this could be covered in a week. And so to explain how this will work, let me show you, first of all, the schedule of the program, and then I'll show you some examples of what the actual training will look like. So the first link that we will go to is the program link for Transcriptomics program. Okay, so please navigate with me to this link. This is where the overview of the program sessions and the schedule is found. So you can see here a very short overview of the whole program. It's a video, and you can also see right here the different topics that will be covered and their schedule. So right here, you've got the calendar, and you can see where those sessions are found. Now here you can also see some of the supporting kind of resources that we are going to be providing you with, which includes the training, the asynchronous coursework that you can look through, mentorship. So I will introduce you to one of our mentors in just a minute. You will be able to collaborate with our team as well as between yourselves as groups. You will have access to big data processing and analysis tools on the cloud. So you don't have to worry about what kind of a laptop you have, or maybe you don't have a lot of resources on your own computer, as well as project support. So we'll introduce you to some curated data sets, and we will give you some guidance on how to analyze these data sets in an effective way. Now, down below, you see here just a couple of testimonials of people that have previously completed our training. So you can kind of see what those people have been able to gain out of this program. And we also are very focused on making these programs as affordable as possible. So this is already compared to industry level pricing is very affordable. But even on top of this, we are providing specific scholarship opportunities for those of you that are interested to join and are really dedicated to learning the resources. So if Mohit, you could also share the link for the scholarship application. Those of you that want to receive a financial assistance and scholarship, we do have those available as well. Down here, you will find some of the coursework that is actually included in the synchronous resources that we will provide. And just to give you an example, I want to go to this link, which is a very introductory course that you will find on called Bytes and Molecules. And so this course right here, I'm not going to play the full thing, but you will see here that there is a very gentle introduction to some of these complex topics that many of you might be familiar with, or maybe you have heard about it, but maybe you don't have enough kind of a technical overview of what all of these terms mean. So we have a number of very introductory courses, as well as some of the more advanced courses, like for example, you can see this project on liver cancer from the Cancer Genome Atlas using big data. Okay, so let me just get to it in a second. Down here, you will see that as a result of completing some of these resources, you will gain a certificate of completion or a certificate of excellence. So the certificate of completion is by just joining all the sessions and performing the assignments that you are going to be getting during those sessions. A certificate of excellence is going to be given to those of you that are able to develop an independent project and showcase that project as the result of your work. And so you will have special recognition and a feature of the program so that you will gain a lot more exposure. If you're looking to advance your career, showcase what you are able to do to people from the industry or from academia, this is what you want. You want to demonstrate that you're able to find an interesting topic in a project and perform the analysis independently with a mentor so that you can identify some meaningful biologically interpretable insights in this kind of big data. So that is an opportunity for those of you that want to stand out. And to help you achieve that goal, we are going to provide you with everything that you need to get started and grow, which includes analysis of big data. And so that's what I want to come to next. Okay, so let's all go to our big data analysis platform, which is called server.tbioinfo. Okay, so this is a platform that allows you to have access to over 5 terabytes of storage, and you can analyze data with a lot of the publicly available data sets using high performance computing. So you can actually leverage some of the computational tools and infrastructure on the cloud, but run those same methods you're probably familiar with. Now, all of you that have registered on the portal can actually perform this demo that I want to do together with you now. So please let me know once you're on this link, server.tbioinfo. So let me know by putting something in the chat so I see how many of you are here. And if you created your account, you will be able to use the same password that you used when you were registering on this portal. Okay, so here on this platform, it's the same password as you have on the Omics Logic portal. Okay, so I'll just give it one more minute. How many of you have already joined this portal? You can now log in. So once you're logged in, you should be able to see your email address right here. That shows you that you have been registered and you have access. Okay, so I know that we have at least 20 people today that are here that have registered on the portal. You should be able to have access. So once you're there, please put in something in the chat box so I know that you're with me. All right, excellent, Jemai. Those of you that are just registering right now, you can actually hover over some of these sections right here to see what they combine, what they offer. You can hover over these eyes. Okay, great. So we have 4 or 5 people ready. All right, great. So here I will show you just a couple of examples so that you understand how easy and fast it will be for you to get started with this program. So those of you that are here, you can see that RNASeq chip is one of these top sections right here. You can press on this drop-down arrow, and we will take one of these demos to show you how the pipeline actually works. Okay, so let's take this first one, demo RNASeq cell line project. This is one of the projects. I'll kind of explain it in just a minute here. And you will see that the pipeline consists of some of the tools that you might be already familiar with: STAR, BWA, Bowtie, Sailfish, HTSeq, DSeq2, etc. So each button here represents an algorithm If it doesn't have anything on it, it's a publicly available script that you can download somewhere else. If it has this little tau sign, that means it was developed at the Tauber Bioinformatics Research Center, and you will find some explanations about these as we go along. So you can click on Start. Here you'll see an overview of what the RNA-seq pipeline actually seeks to do. It first starts with removing technical artifacts from the short sequencing reads. Then we will perform mapping onto the reference genome or transcriptome. And after that, we will quantify the expression levels of genes and isoforms to understand structured data, to get structured data that we can do downstream analysis on. Once I've clicked on the first step, I now have different buttons highlight. That means that my selection is limited to the buttons that are highlighted. So let us make a very quick pipeline. We will do some pre-processing using Trimomatic. What is Trimomatic? It trims technical elements from sequencing reads, preparing them for mapping. So before we do mapping, we eliminate those sequences. So here you click on OK. Then we'll actually eliminate bridge amplification using PCR. So this is some kind of a technical step inside the sequencing process that could result in technical artifacts for our downstream analysis. So we want to eliminate that for highly expressed genes. After that, we can map our prepared data on the reference transcriptome. Now, how many of you can tell me what is the difference between the genome and the transcriptome? Okay, those of you that want to become mentors, this is your opportunity to show us how much you know about this. What is the difference between the genome and the transcriptome? Okay, Harshita is saying transcriptome is mRNA, genome is the entire DNA. Excellent, great. So Harshita, talk to us after this if you want to be a mentor. You've got an opportunity. You can send us your resume. So here is what we will perform is mapping on the transcriptome, which means that we want to analyze how reads map onto the regions that are already present in the expressed genes and isoforms. So this is going to be using the GTF file that contains the beginnings and the ends of all of the exons that are spliced together to produce a transcript. So now we perform this mapping, and after that, we want to prepare a table of expression. Okay, and so what is a table of expression? A table of expression is a matrix that contains gene expression values, but in a data frame that contains the header, which is the names of our samples, which we can organize into groups by phenotype, as well as genes, which are typically going to be in ensemble format. So here we will perform this RSEM expression table, and then we can just simply click on End, and you can see the full process from beginning to the end. And now you have also some available outputs right here. So let me pause right here and let me answer some of the questions that I've been mentioning and I've been seeing in the chat and I didn't have time to address. Okay. So first of all, what type of tools are we going to be teaching? So all of these tools that we use right now are open source tools, and they are hosted on the cloud. You don't have to use them on the cloud. You can use them on your own computer if you would like to, but we are going to be, for the sake of time, using the big data processing side here on the platform. Just after we complete this, I will move on to the coding part so that you can also appreciate the training in R and Python that we will also include in this program. Okay, so hopefully that answered your question right there. And I hope that the person that had a question about the schedule, you will first of all hear more about this. And then if you have any questions, then we can answer that as well. Okay, so now we have our table of expression. You can see that we have genes and isoforms. So once we have this data, it's structured data. It's not large data sets anymore. This is condensed now. We have a lot of features, but we don't have to process individual reads on a huge genome, right? So this data has been prepared. So what can we do with this type of data? Let us now go to our Learn platform. So you can go here into this section just by clicking on courses. And I will show you what you can start doing with this type of data to gain insights about how gene expression can show us differences between phenotypes. So to do that, we can go to a course called Transcriptomics. So if you just type in here, Transcriptomics. So you can see here in Transcriptomics that all of the detailed steps on how the data is generated, what are the objectives of sequencing, how do we process and analyze this data, what are some of the statistical tests that are being used, and all the other topics are going to be covered here. And then you will get an introduction to some of the methods for downstream analysis of this type of data. And so one of the things that I want to take you to is now a tutorial on differential gene expression. So I'm just going to put this link here in the chat box as well. You will see that now that we have the table of expression, we need to learn how to organize our data into a data frame that is ready for statistical analysis and visualization using ggplot. And so here you will see how we create a data frame, how we place it into an object, and then how we can plot different plots to find differentially expressed genes. And the cool thing about this is that you can actually run this code right here in your browser. So you don't have to necessarily install all the different kinds of RStudio and the packages on your computer to know how this works, right? Because one of the major objectives here is to understand where does this fit in to this overall scheme of things. How do you actually use differential gene expression to get a biologically interpretable result that you can speak about in terms of pathway analysis and biological function. So right here, I can run this right here in the in this little practice console. I will get my output. So you can see that I have an output. This has completed, and you know whether it was done correctly or not. So here we have a very small output. We can look through and read our code to understand what it is, and we have a plot that shows us differentially expressed genes, as well as provides us with a table. And in other tutorials throughout this course, you will find how to do different types of visualization for this table and how to do downstream biological interpretation. So now what I want to do is, once you've understood how this works, we hope that we have eliminated any questions about how so quickly we can get from learning about the next generation sequencing technique to then being able to analyze and then being able to apply different statistical analysis tools as well as biological interpretation. I want to actually introduce you to one of our mentors to speak about her background so that you will appreciate the kind of support that you will be getting as you go through this program. So I want to pass it on now to Dr. Harpreet Kaur. She's a bioinformatics trainer. She has a PhD in bioinformatics, and she will be one of the mentors in this program. Harpreet?
Thank you, Ilya. Hello, everyone. My name is Harpreet Kaur. I have a PhD in bioinformatics. Let me give you some glimpse how bioinformatics can be helpful in identifying signatures for a specific disease condition. Like here, I'm giving example from my doctoral research work. So my PhD research work was on biomarker discovery for the diagnosis and the prognosis of the liver cancer using bioinformatics. So, but inspired me to study the liver cancer. So it's the high mortality rate of this malignancy and the lack of precise biomarker that lack precise biomarker specifically that can diagnose liver cancer at an early stage. Worldwide, liver cancer is considered as sixth major cancer in terms of incidence and third in terms of mortality. Often at times, liver cancer is detected too late for an effective treatment. So another factor that contributes to poor outcome of the liver cancer patient is the frequent reoccurrence of tumor after the treatment. So with keeping these concerns in mind, I have framed different objectives that can address these concerns using bioinformatics techniques. For example, in the first project, we have used large-scale transcriptomics data to identify a diagnostic biomarker panel for the hepatocellular carcinoma. So in this study, we employed 30 datasets containing nearly 4,000 samples to elucidate transcriptomic signatures for the diagnosis and the prognosis of the liver cancer, employing various statistical and bioinformatics techniques. In the second project, we used transcriptomics and methylation data to identify a biomarker panel for the stage classification of hepatocellular carcinoma. Here, we use TCGA data of 350 samples and elucidate 51 features, and that include 30 RNA transcripts and 21 methylation CpG sites using number of data mining techniques. In this study, particularly the naive-based model classified early and late stage sample of SCC with nearly 79% accuracy. In the next part of the project, we use transcriptomics data to scrutinize diagnostic biomarker panel for the cholangiocarcinoma, which is another major liver malignancy. In this study, we employed 9 datasets containing 350 cholangiocarcinoma, 130 adjacent non-tumorous, 90 SCC samples to identify cholangiocarcinoma specific transcriptomic signatures, implementing number of machine learning techniques like extra tree, SVM, random forest logistic regression, etc. So in conclusion, we have identified a universal platform independent 3-gene based biomarker that can predict SCC patients with high precision and also possess significant prognostic potential. Then we identified a 3-gene based cholangiocarcinoma specific transcriptomic signature that are capable to discriminate CCA from SCC and the normal samples with nearly 90% accuracy. At last, we have identified a biomarker panel of 51 features that include 30 RNA transcripts and 21 methylation CpG sites to predict hepatocellular carcinoma at an early stage. So in my current role, I am curriculum developer and research consultant with Pine Biotech. I'm assisting participants to learn about data analysis, machine learning, and visualization in application to large data sets used in the biomedical research. So to provide hands-on training and to make participants familiar with the approach to design and complete research projects, we are continuously designing different research projects using publicly available data based on the studies published in high-impact journals. Thus, we have a collection of research projects where you can learn how bioinformatics can be applied in the field of different diseases, including oncology, infectious disease, based on the publicly available data to drive biological and clinical features from that data. So for example, let me give you one example, one of such project. Here, we have explored different omics layer, that is RNA, miRNA, and clinical and demographic data, employing multi-omics data integration approach for the liver cancer patient to find out molecular signature that are associated with the clinical phenotypes of the liver cancer. As one of the major challenges with the malignant conditions, that a single omics layer is not sufficient to design highly efficient biomarkers. Thus, multi-omics data types can fill that lacuna and will facilitate to design efficient signatures. But then another major challenge, the integration of those multi-omics data types. Either you need to develop an individual pipeline for each data type, then you will find an approach or a method that can integrate them together. So for this, you require either highly computational or a bioinformatics skill. So to remove these obstacles, various multi-omics data integration tools like SNF, iCluster Plus, O2PLS are integrated on the T-Bioinfo server, where users just need to provide input data set and can easily perform various types of multi-omics analysis by just clicking on options like already Ilya has shown you. So in this project, we have also implemented these multi-omics data integration tools. So finally, we are trying to understand how multi-omics data integration analysis can provide us a holistic picture of this lethal malignancy, and also how they are associated with the diverse clinical phenotypes like tumor stage, risk factors, race, age, gender of the patients. So participants can explore this project during the upcoming program to get a better understanding. So beside that, there are a number of the research fellows who have completed their research projects in the field of oncology with the Pine Bioinformatics Research Fellowship Program. So the first one, Miss Urja Parekh, who is postgraduate student from India and completed her research projects on identification of biomarker to differentiate between neural cells and glioblastoma, employing publicly available data using different tools that are integrated on T-Bioinfo server. So our another research fellow, Simayet Dolanar, who is an undergraduate student from the Turkey, recently completed her research project entitled Long Non-Coding RNA as Therapeutic Target in Chronic Lymphocytic Leukemia, using publicly available data and integrating various tools from the T-Bioinfo server. Then we have Kala Umari Marul, who is a PhD student from the Mexico City and completed her research project entitled Classification of Triple Negative Breast Cancer Cell Lines by Gene Expression Analysis of the Transmembrane Protein using Machine Learning Algorithm based on the cell line data, employing multiple machine learning tools that were integrated on a T-Bioinfo server. So I'm looking forward to being your mentor in the upcoming Transcriptomics program. Now, I would like to pass this session to Ms. Sonalika to talk more about this program. And if you have any question, you can please go ahead and post your queries in the chat box. So thank you. So over to you, Sonalika.
Thank you, ma'am. I'll be just sharing my screen in a while. Yes, thank you, sir. Good evening, everybody. I'm Sonalika Ray. I'm pursuing Masters in Bioinformatics from Punjab University, Chandigarh. And currently, I'm a research fellow at Pine Biotech. My research interest lies in omics data sciences, computer-aided drug designing, and pharmacoinformatics. So today, I'm here to share my path to computational biology and how bioinformatics shaped my career. So as a student, it all starts in 12th. Must have happened with all of you, no doubt. So I took medical, and like any medical aspirant, my only aim was to get into MBBS. I worked a lot hard, but destiny could not get a government seat. I had I had heard a lot about bioinformatics as the most promising field, and the subjects gained my attention, and I got a seat for pursuing bachelor's in bioinformatics honors at Punjab University. I learned a lot, gained a lot of insights and about many fields, and then came the time for my master's. So I started exploring about more fields. I thought, let's explore some other fields in the master's, and somehow I started thinking about other fields like stem cell engineering, genomics, genetic engineering, or the biomedical sciences. But then my professors made me realize about how vast bioinformatics is, about how it comes under all the domains, like how it covers all the other domains, and how pursuing masters in bioinformatics could help me do research in whichever domain I would want to. So that made me pursue masters in bioinformatics. And then came my second year of masters, and I was very much interested to take up some or the other internship program and do some sort of hands-on analysis or learn more about how actually industry works and what skills are required. But what happened in my second year itself, COVID happened. So I started looking for opportunities online, and I was fortunate enough to find out the Pine Biotech website. So I found a great deal of courses over there. There were many projects over there. And also I was surprised to see that they had got the R and Python code integrated in their server itself. So I took up the courses, I learned a lot of them. And then in January, I was fortunate enough to get a research fellowship, and I started my research project, my master's dissertation project on liver cancer with them itself. And just in the span of 3 months, I was I could finish my project. And academics has designed the program such that we need to take 6 months to do our project, but just with the help of the server, I could do everything in just a matter of few months. So many a times people actually ask a question why bioinformatics and where it stands today. So actually, I just wanted to discuss how important the field bioinformatics is. So bioinformatics is actually the collaborative study which integrates a lot many fields. So it has biology, computer science, maths, biochemistry, statistics, chemistry, engineering, and data science. Obviously, that's the most upcoming study right now, right? So it's not like that bioinformatics has no scope, or it's like a smaller part of some or the other field. But actually, what I think, it is the father of all, because all the main branches, they come under what? Bioinformatics. So getting a hands-on and getting yourself trained with all the skills of bioinformatics is like the most important thing right now. And as we all have been discussing that in our academics, what we face is a lot of theoretical part of concepts, because our syllabus, first of all, doesn't get updated timely, and it focuses more on the theoretical part. So this is what I'm talking about, the Indian education system. I'm not blaming any system, but yes, it's like that. So yeah, we need to actually look for more of trainings, and we need to train ourselves with the upgraded skills. So that's where Pine Biotech helped me a lot. So first of all, my research project, I was very much confused which topic to take, because it happens with almost each and every student, I believe, that whenever our projects are about to start, we think, okay, which topic we should start with, what to do, how to do, and things like that. So the mentors at Pine Biotech, they were so pretty helpful to me. Like they helped me with what topic to choose. So Ilya sir, Harpreet ma'am, and Mohit sir, they actually helped me with my topic. What should be my research question? They helped me frame each and everything. And they were with me at each and every point of my research project. Whenever I faced an issue, they were there to help me. So mentorship, it's amazing. And then the best part is that we don't get such pretty big labs or the instrumentations which is required to carry out the research at a great level. And I used to believe that just because my institution can't afford these expensive labs and machines, that means that I might somehow lag behind others. But no, actually, Pine Biotech provided that server to me to carry out whatever analysis in the world I can think of, right? So all my research project analysis was through the Pine Biotech server. And what I did in my project was I repurposed drugs based on the transcriptomic data of hepatocellular carcinoma cell lines. So I needed to carry out a lot many things, let it be machine learning, or to find out the differentially expressed genes, see where the clusters are present, And also then I had to integrate a lot many visualizations to understand my results in a much better way. So that's where TBioInfo came to my rescue. So yes, it's a great platform. And moreover, if you're interested to learn more about others' projects first, because many a times if you are a non-bioinformatics student, you might be confused about what to do and like what the topics are, like to know more about the existing opportunities, right? So there are a lot many projects integrated on the server which you can like go and have a look at and understand what they did, how they did, what was their methodology, and the research process. Yes, and also there are many like demo pipelines which you could follow. And moreover, the best of all is that there are like the bestest of the trainings and webinars where each and every assistance is there. Like you can see yourself today itself, like each and everything was told, though not in that much detail because yes, there's a lot to cover, so we need time to do that. But yes, up to a great extent it was. So yes, I feel myself fortunate enough to be a part of Pine Biotech, to have done my master's dissertation with Pine Biotech even in the COVID times. And yes, so I thank you all. I thank the team specially who helped me a lot to complete my project. Thank you. Now I would like to pass it on to Dr. Mohit.
Okay, thank you, Sonalika. And also would like to thank Harpreet and Ilya. So there was a question on variant analysis COVID-19. So I wanted to share this. Maybe I'll just go back to the presentation. Okay, so hello again. My name is Mohit Mazumdar, and I'm one of the global representatives of Pine Biotech. And in my current role, I'm collaborating and working with academia and industry to provide support for bioinformatics research and education. So along with our hardworking and efficient team, we have been able to work with more than 30 universities for the past couple of years for research-based programs and helping thousands of students to develop understanding of data-driven research and life sciences. So in this program, what we'll be doing is that we will be utilizing some of the top rated online tutorials using the Learn portal. This has the smart tracking as well as curated data sets designed for great learning experience. So after that, to tell you about the program. So in this program, we will combine online sessions, self-guided study material, and practical assignment for an immersive experience that has proven to be very effective in our well-known and respected omics logic programs. So we will learn about important concepts within biomedical research, such as infectious diseases, cancer research, and learn about immunology of infection, molecular disease markers and stages, and how these diseases can be diagnosed and treated. So we will be talking about several bioinformatics methods, such as processing of RNA-seq data and learning about differential gene expression and more. And finally, we will look at the sample projects and learn how to create our own projects with the bioinformatics method using the public repositories. So by learning about these major concepts, you will be able to equipped to start your own project. So about the schedule, so the upcoming program, we already have seen the schedule, and we'll go back to the page where you can download this entire schedule and more descriptive one. But for this presentation, I'll just take you through the sessions really quickly so that you'll understand how this is, how this is going to be. So the upcoming program will focus on NGS transcriptomic data analysis that include performing downstream analysis and applying data science techniques and machine learning methods. So we will have almost, 4 weeks to discuss the various aspects of RNA-seq data analysis and how this field of bioinformatics is continued to evolve and amaze us all. So to start with, the first session will be about, learning about mRNA library preparation, mapping on the transcriptome using algorithms such as Top Hat, HiSet, Bowtie 2G, sailfish. So we will be discussing and telling you that what this algorithm does, and we will show you how to run this algorithm, so what to expect from that analysis. So that's why I think T-Bioinfo platform plays a very important role, which was discussed earlier. So the first session will be on 19th May, and we'll be looking forward all of you to join the session and learn from us and learn from the resources and come up with a project and do your internship, all of that. So the second session will be about processing transcriptomics data, which will be on 21st May. So here we will be discussing strategies of mapping exon and junction detection, and then techniques such as normalization and quantification. And this is all covered in Transcriptomics 1. So the Transcriptomics courses will be available for those who are joining, and along with the projects. But this course will give you a great overview of how this entire NGS analysis is. You can start and expect results. So after that, on 24th May, we have the session on project, and also it is more about reviewing. So so we will be reviewing a publication. So as we said and discussed, and Dr. Harpreet also showed you that this is a more of a research-based program. So in this session, we will be learning about modeling precision medicine. So here the example is breast cancer cell lines and the application of machine learning. So this is a paper by Demian et al., modeling precision medicine for breast cancer. So this paper talks about 70 different breast cancer cell lines and 90 different therapeutic agents, so drugs. So this project also include SNP data, so microarray data, RNAseq data, transcriptome, and then exome and genome-wide epigenomic data as well. So our focus here will be to understand about precision medicine that actually revolutionize the way patients are treated by using precise molecular information that provides reliable indicators on the effectiveness of the treatment. So I mean, that is one of the major objective of the program. So in this project, we will when you will be first learning about the publication and how they did it, and then we have a project on this publication. So in the project, we will leverage cell lines from human breast cancer tumor and study that how they integrate with the other omics data and the response to various cancer treatments. So what is the response to the drugs? So as a result, you will learn about various omics data types and how to integrate them, associate them with phenotypic changes. So the response to the treatment would be the phenotypic change here. And we will be using sophisticated machine learning algorithms, obviously, because it's complicated multidimensional data. So after that, on 26th, we will be learning about loading these data sets into the Python and R, and then how can we use that and prepare matrix and data frames and create variables and stuff like that. So after that, we will be learning about normalization and preparation, like what we can do for to create a pipeline and what we expect from the results from that pipeline. So also like removing zero, non-zero and log scale transformation. After that, we will be learning about how we can do this like and see visually. So doing the box plots, scatter plot, histograms, heat maps, after the transformation, doing it on the coding side of things. So after that, we will be learning about exploratory analysis in R and Python. So here we start about PCA. So from descriptive statistics to machine learning. So here we will start about exploratory data analysis, and we will be using coding as well as the T-Bioinfo platform. And then one of the major objective of this program is to learn about how the changes in expression affects the phenotype. So differential gene expression is one of the tools that is used for this. So we'll be learning about DSEQ algorithm, EdgeR, and volcano plots, and all of that. So that would be on 4th June. And then we have about the next part as regression analysis. So here, because we are doing comparison, we are making groups, and then groups have different characteristics, features. So we will be learning about t-test and ANOVA regression and factor regression analysis to get into deeper insight of regression analysis. And this is also very much covered in transcriptomics too. So even if you miss the session, or if you want to prepare ahead of the session, so you can do that, so that you can make most of the session. So this will be also available for the participants to do the courses first and then attend the sessions. So that could also be done. And then we will learn about biological interpretation and in the context of single cell. So how you can do single cell analysis and how you can come up with, KEGG pathway annotation for differentially expressed gene, understand about the biological process, molecular function, and all of that. So by this time you have learned about the different techniques and how you can apply this to different kind of data sets, and also how to understand the biological interpretation, how to interpret the biological how to connect it with the biology. So after that, the next step is to learn about the application of this technology into different areas of research. So the next session on 11th June is about that. So here we will be discussing about literature review, compiling a data set, developing analysis plan, and doing that entire transcriptomics data analysis, and how that is applicable to neuroscience, infectious disease, and oncology. So we already have these projects, and we already discussed about molecular cell precision, molecular cancer, modeling cancer precision medicine, but we also discussed the TCGA project, and we also have the projects on neuroscience and other projects that you can learn from. So during this time, we will be covering all of that. And as Ilya said, we will be utilizing these platforms and resources. So quickly, just to tell you that this entire Omics Logic programs will be utilizing the Learn platform, where you have created a login. So I think if you have not, please create a login here. It's a free login. So I mean, you don't have to join the program to create a login, but you can, again, if you're not joining, but still you can avail a lot of resources and learn from the resources. And maybe once you're more comfortable, you can think about what training you would need. But if you are interested in learning this, so I would recommend you to join this program now. It's a great opportunity to join this, because we have multiple collaborators and different expertise, and we'll be covering a lot for this summer, because summer is the time where you will have time and we will also have time. So here, the second platform that we'll be utilizing is the TBioInfo platform, so which you saw, and the third platform would be the coding platform. And then we will be applying it using machine learning, multivariate analysis. We will be using data mining and classification algorithms such as hierarchical clustering, decision trees, random forest, support vector machine, and also SVM, and support vector machine, LDA, linear discriminant analysis. So we have all of them on the platform. And then we will be practicing the data science tutorials using the code platform, and we'll be applying that for visualization and downstream analysis. So this will be the part of this entire program. All of the access will be the part of the program. So you will be getting access to the different platforms, as well as the tutorials from where you can learn on your own from your browser, because you don't need to even install these programming tools and also like the softwares. So out of this entire program, you will be able to learn and understand from the literature, and you'll be able to understand the steps that are required to do the NGS data analysis with reproducibility. So there will be a constant accuracy, and you'll be guided by the experts who have worked throughout their career and knows in and out most of the things. But obviously research is challenging each time you get a new question. But however, like, we have quite a bit of experience working throughout, as Ilya said. So we'll be helping you do these steps, understand from these steps, and try to make the most from the learning that you get. So in this entire process, we will be learning about R and Python, as said, statistical techniques, how to manage and analyze data, develop communication skills about how we can present this information that we have created by the form of a slide deck, by the form of a video, or by interacting with us like in one of these sessions. So the sessions that we will have are with the participants and with the trainer. So you'll have a lot of opportunities throughout that time, hours of time, to discuss your own problems and come up with solutions. So we'll be doing all of that. And to registration and downloading all of this at one place. So let's just go again to this site, the Transcriptomic site that we all went before, and then learn about the process of the registration. So here I have the site open. So if anyone can share the link, so let me share it. Yeah, there you go. So this is the site for registration. If you have already logged in, so your name will come up here, your name will reflect. So once you're here, you can request for more information, like joining a webinar like the one you're doing, and you can also start your application. So once you start your application, you come to this place, beginners, intermediate, and advanced. Now you have three different levels to choose from. So in intermediate, in the beginners, you will get all of this that we discussed today. The difference between intermediate and beginners is that here you can request for special one-on-ones mentor support to help you with any of your questions. And if you feel like that you need additional support and that would be good for you, so that option is there on mentor on this intermediate option. And then for the advanced option is for someone who would want to work on their own data and who would want to work on using the TBioInfo research platform. So the research platform enables you to upload your own lab data, which is not publicly available, and you will get a research license to run your analysis and get support for that. So that's the research license, and then you can sign in and check out. So in addition to all of this, till tomorrow, there's a special code that you can apply. So you can get a 30% discount on all of the above. So the registration fees will be reduced. The scholarship is 30% for all of you, which is an early bird scholarship. So you can apply that 30% scholarship, and this will be available for $105, which is around 700 rupees in Indian currency. So please feel free to let us know if you face any problems. So I'll just show you that how you can apply that maybe quickly. So I'll start with logging in, and once I log in, I will be able to apply it for the payment process. So please feel free to let us know if you have any other questions related to what we covered today. And again, I mean, if you have not joined the groups, please feel free and go and check the groups here, the group on LinkedIn and the group in Facebook, where we keep on posting about our upcoming programs, free webinars, and a lot of resources, free resources and good resources that you can utilize and learn from. So let me just check if I have it open, because actually I don't use Safari, so maybe I can show you here, or maybe you can let us know in case you feel any problems, while doing this. But anyways, please let us know if you have any questions, and we will be happy to answer those now before we end this session. So yeah, Ilya, are you sharing? Because it's not loading, I guess. Yeah, so here you can click on Buy Now, and then you get this add promotional code here. Yeah, so NGS 30. Yeah. Just a second. Let me confirm that.
Well, in any case, maybe you will email the code for those that are interested so that they can do it.
Sure. Yeah.
Okay.
So if there are any questions, please let us know and we will be able to answer those. So is there any possibility of doing postdoc in transcriptomics based drug designing? So yes, I think BSS, there are a lot of opportunities that are available on this sort of training and projects. So I see there are a lot of projects on transcriptomics data analysis. And also, like, if you have much experience, a little bit of experience of working on these data sets yourself and exploring that what are the challenges that are there. So those are the questions that you get during the interview. So it is better to get a hands-on feeling of how this data is applied to different kind of researches, because it's just not like this, but it has, like, the omics data or bioinformatics has a lot of application in all of the fields of areas of research. So I think more one or now or then it will be useful for you if you can do that. So yeah, I mean, just let me, we have emailed you with all of that information if you have registered through the link, but just like give me 1 second, let me confirm the code with all of you so that you can apply that to this, because the code is going to expire tomorrow. So yeah, it's NGS30. I guess we will be checking it and emailing you again. So please look for emails from us. We will be emailing you from marketing@pine.bio, and you can also email us if you have any other questions at mohit@pine.bio and marketing@pine.bio. And Sonalika, if you can write your own email address as well. So please feel free to reach out to us and let us know if you have any problem. And thank you for joining today's session and be here with us and listening to us. I hope that today's session was useful for all of you. And with that, I would like to conclude today's session, and I hope to see you on 19th. Have a great rest of the week. Thank you, everyone.